So my name's Stuart MacGowan, and I'm in Jeff Barton's group in University of Dundee in Scotland. And we're not virologists, but we're structural bioinformaticians. And the last couple of years, we were contributing to the COVID-19 effort. And I'll tell you today what we've been working on. Oh, I'm just gonna have. Okay. So the talk, I'll have a little bit of an introduction. And I know you all know what COVID-19 is, but I'll just some context. And then I'll tell you about our expertise in the lab, just so you know why we got into this and why we were well suited to do what we did. Then the results, the results and kind of current future work will be split into three parts. So the first part is stuff that we did really, really quickly in 2020, purely computational to address the kind of ACE2 genetic susceptibility hypothesis. And then in the next part, we had collaborators come in, test some of our results. I'll tell you what that meant for interpretation and for what we thought about the predictors. And then I'll tell you what we're working on just now, which is to try and see if there is direct clinical relevance, and also looking at the interplay between Spike and ACE2 variants. So yeah, what can I say? I mean, some of these numbers, confirmed cases, like discounting reinfections, that's like 7% of Earth's population. Yeah, the deaths, the economic toll, we lived through it. We're coming out the other side of it, but researching this is really important. It could happen again, and it's not finished. And there are other diseases as well that we can, the scientific community really rallied. Like if you search ACE2 Spike on PubMed, it's like 6,000, and that's just like one part of COVID research. Like it's incredible. Okay, so what we do in Dundee is Jeff Barton, he's like a well-known figure in sequence analysis and structure prediction. Some of the biggest hitter stuff is JALView, which I hope you all know. It's like a fantastic multiple sequence alignment edit and visualization analysis software. JBread 4, which was a great secondary structure prediction algorithm, but we're looking at what that means in the context of the AlphaFold. And the Dundee resource for sequence analysis and structure prediction, which is a set of diverse tools for function prediction. Now, in more recent years, we were focusing on human genetic variation in protein structures, and this is what I've focused on. And we were trying to find out how, human genetic variation, protein structure, and multiple sequence alignments, how you can integrate those data sets, and how you can use population variation for things other than pathogenicity prediction. So we were interested in that too, but we were also thinking about like structure prediction from human genetic variation. Okay, so in 2020, before we had vaccines and before we had variants, we had people, we knew a set of comorbidities, age, sex, pre-existing medical conditions. But the big question was like, what other factors, why are some people getting severe COVID and others aren't? So one of the a priori candidates was ACE2, which was the cell entry receptor. So people thought, well, variants in ACE2 could affect infection. So what this is what you see when you load the ACE2 sequence in JALView, and you map uniprot features and nomad population variants to the sequence and the structure. So on the sequence, the nomad variants are pink, and on the structure, they're magenta. And I'm just showing the ones on the structure that are at the spike interface. So ACE2 is green and spike is kind of tan color. And seeing this, the immediate question is like, could ACE2 variants affect binding? Sure, they could, right? But, then you're like, can we predict how quantitatively? And now we have expertise in machine learning. So we know that when you ask that question, you also have to ask, how can you trust the predictions? Okay. So, I did a Google. There are methods. There are people in this room who made the method that I landed on for predicting the effects of mutations of variants on protein interfaces. And they performed well in various metrics. And like I did look at a few and I have results. I'll just focus on one. But MTSM PPI2, had great performance metrics, a web server that I could use. So I got the predictions, right? And this is just a sample of the variants here. But it was easy to get the predictions, but how do I know I can trust them and how do I know what they mean? Now, fortunately, in 2005, there was a data series of ACE2 mutants that were assayed against the SARS-CoV-2 spike. So I could use that to benchmark MTSM2 PPI2 in this system. And I checked that this wasn't in their training data. This was old data, right? They hadn't trained on this. And what you can see is that the delta G is strongly associated with this binding assay. It's very sensitive, it's quite specific. And what that could let me do is interpret these negative values and also the ones around 0, But what you don't see here are any predictions or variants that enhance affinity, right? Because there weren't any in the data set. So I don't know, I didn't have any confidence in that. Now, the original authors used a clever trick. They exploited the fact that delta G is a state function. So the reverse mutation has to equal the negative of the delta G for the forward mutation. So I modeled the reverse mutations of all those to use as a benchmark. And what you can see is that it's less specific, it's less sensitive and, less specific for non-binders, and it's less sensitive and less specific for predicting enhanced affinity, right? Not altogether surprising. Now, we looked closely at each individual variant that was like pulled out by this method to kind of structurally validate it. So we kind of thought that the 1 kcal/mol threshold, whilst not as good as the inhibitory variants, it gave an indication of enhanced affinity. And you'll notice that I wouldn't claim at this point that would mean a risk factor, because that would be unclear here. So, okay. Oh, sorry. Yeah. So just to give you some context for that work, that was in April when Jeff emailed and was like, can we look at this? That was like 1st of April, and we submitted the preprint to bioRxiv at the end of the month. It was pretty intense, 18-hour days in a 1-bed flat with my wife and a toddler. Yeah, I owe her everything, but I would not do that again. Okay, so what happens when someone tests your predictions, okay? So we found collaborators who could make this, who could make ACE2 mutants and assay the affinity. And at first, they did 4 mutants. So we picked 4, we picked 2, D355N, the top 2 were ones that we predicted would strongly inhibit binding. And so we were confident in those. And the next 2 were predicted to enhance affinity. So we were like less confident, but really interested in those. And what you see is like, I've said that the negative ones were confirmed. You only see 1 here because D355N just didn't bind at all. So that was a good prediction, but you'll see that the positive predictions were not good. Like they were totally wrong. So this led to a reevaluation of what we'd done, but they did more for us, which wasn't quite so bad. So at first, when the guy came back, actually Michael Barton, Jeff's son, who did the experiments, and he said, oh, you've only got 2 out of 6 of the correct class. But when I had plotted them, I was like, okay, but these new ones, we've got the right rank for 5 out of 6. Okay, so like bear with me for a moment because you might scream when you see the next plot. If you exclude what I'm going to call categorically incorrect predictions, you see a really strong linear correlation, like a very, very significant association. And now I'll show you some orthogonal evidence of why this is probably okay, but I actually think that the predictor has made a category error for those variants. It's not just got it wrong about quantitatively, it's convinced that they enhance the affinity, and then it's like going, oh, I wonder how much by, But these 2, maybe we can correct that offset. Now there's some there was deep mutagenesis screening data from Eric Prockel and co-workers, and this is comparing the deep mutagenesis screen to it shouldn't say recalibrated, that's the raw score. And what you'll notice is that, when you've got a very negative prediction, you tend, so below the line on the left, they're concordant. But when it's between 0 and -1, it's kind of a bit all over the place. But you'll notice that they're like kind of all squeezed behind 0, right? Now, when we apply the recalibration, nothing happens in terms of what is going on below -1. But I actually think this is a better representation of the ambiguity of around that. And not only that, you get a little bit improved sensitivity at the 1 kcal per mole threshold, right? Now, I actually looked at you guys' training data, and I think this offset is in there. So I think it's a global thing. At first, I was thinking maybe it's just a system that I need to correct for, but it might be a global thing in the method. But anyway, how do we relate affinity to infectivity? So there's some things that are obvious: no binding, no infection, unless there's an alternative pathway. Okay, and in SARS-CoV-2, to my knowledge, I don't think there is. You can find a paper that says anything in this field, to be honest. Like, so yeah, I have seen some other alternatives speculated upon. When it comes to lower affinity, you've got kind of two questions. You're like, do you get a proportional reduction in infectivity, or could there be a threshold below which you get no infection? And I mean, from the literature, we in 2020 we had some hypotheses about this. For higher affinity, as a risk factor, no effect is unclear. There's mechanisms that you could envisage that could lead to more risk, but it's certainly not as clear cut as for inhibitory variants. Now, during review, we got turned on to a paper where they'd actually looked at infectivities of ACE2 variants, right? So whilst that's a fantastic resource for interpreting the variants in its own right, we could also use it to now draw a line between affinity and infectivity. And I think you'll agree, I mean, this kind of answers some of those hypotheses. So you do get with big inhibitions and binding, and now you'll see this is against the experimental delta G. I've got D355N up there. That's an upper bound based on the sensitivity of the assay. But when you've got really reduced binding, there's almost no infection. When you've got a little bit down, you're getting proportional reduction. Again, it does look like there's a threshold above, and when you get more binding, so that's the Spike M501Y mutation, which our collaborator showed really increases affinity. You don't get more infectivity. Now, this doesn't rule out a biological effect. Actually, there's some evidence to say that because of the interplay between cell surface expression and affinity, it could be that you get different curves and different thresholds for different cell types. So if you have less receptor, then you need there's a higher affinity threshold. So yeah, enhanced affinity, and obviously that opens the way for like affinity lowering mutations that do other things like antigenic escape and whatnot. So yeah, this was fine. So this is the kind of refined insights that took a lot longer because we had experiments going on. So the delta, the prediction straight up gave us really rapid insights, and we were right for some things and we were wrong on others, but we had an inclination. In hindsight, like I kind of really wanted those positive predictions to be right. So maybe I was a bit biased there. But we were able to prioritize experiments as well. And then like 10 experiments gave us massive insights into how well this predictor did in this system. I mean, it answered the questions for these specific variants and the relevance of that to COVID-19, but also let us recalibrate things. And then also we were able to go one step further thanks to an insightful reviewer and draw a line to infectivity. And what this means is, because they only assayed some, But if we can predict delta delta G for all of them, we can map back to infectivity. And I'm not sure here, but they looked at the Proco DMS screen, and we got a better correlation with the MCSM recalibrated predictions than with the Proco data. Okay, so I'll just finish with some stuff that we're doing at the moment. So I was trying to get away from this. And then Jeff said, oh, I've met this guy with a clinical data set. So Kenneth Bailey in Edinburgh, who is now, and this is what I always wanted to happen, is they're going to look and see if there's an association between affinity and clinical outcome. So loads of people were doing burden tests for ACE2, like right at the beginning. But when I was reading these things, I was like, but some of them could have opposite effects, you know, and you're adding them all up together. You need to stratify the variance somehow. And this is a metric that can do that. So that's going to be tested. But what they really need is strain-specific predictions, right? So we have another paper that I've not described here, which was focused on RBD variants, but also RBD variants interacting with common ACE2 alleles. And there is a story in there of non-cooperativity. So when the mutations on each on ACE2 and on Spike don't just add together when they interact, such that like if you carry this S19P allele, you are actually protected with the S477N mutation that went around France and Australia for some times, in contrast to someone who didn't have that allele. So it's really important to give them strain-specific data, and they're going to run a pilot with their experimental data, but we only have limited experimental data, and they have more variants, and they need scores for those. So now I'm looking at predictions, but I need to benchmark whether or not the predictor can identify these subtle interactions. So that plot there is showing predictions from a delta delta G predictions from an Omicron structure versus a Wuhan. I was actually really glad. I really pushed for them to do S477N because I thought it would interact. S477N a couple of years later cropped up in Omicron. So that's why, yeah, that one's highlighted. And then the other thing is like some of these predictions are quite far from the interface. So I really need to kind of figure out how reliable the predictions are. Or do I just use a heuristic and I say, if it's so far away, it doesn't affect it. So I will skip that and I'll go to acknowledgements. So these are the people who contributed. In particular, Michael Barton did all the experimental side with help from his group. And of course, you know, the resources, MCSM, PPI2, NOMA database, PDBE, UniProt. Yeah, couldn't do this without it. So thank you for listening. These are links to the papers, and please come speak to me or contact me. And thank you.
So, how did you go about like choosing these mutations to make this almost linear plot? How did you go about picking which mutations to use?
All right. So actually, once we realized that the predictor didn't give us such great results in our series, some of them weren't chosen on the basis of a prediction. They were chosen on the basis of like epidemiological interest, right? So S19P and K26R had a near zero prediction, right? So I actually kind of never really thought, but they were the most common alleles at that interface. And by that, I mean, they're still really rare, like less than 1 in 1,000, but most variants in NOMAD are like less than 1 in 100,000, right? So that's why those were picked. And then there was a bit of a combination. I did want to get a little range across the predictions. I mean, now you'll see there's a little hole in that correlation that if I could go back to them, I'd be like, oh, can you make one? Can you for a variant that has a prediction that would plug that gap? Like, would you do that one now? But they're on to other things. But yeah, so it wasn't some voodoo. It was like, it was, yeah, serendipitous, I guess.